All right, hello and welcome to our A12 nucleic acids review. We're going to be going over all the different standards, starting with standard 1. So standard 1 says the DNA is the genetic material of all living organisms. And so this is going to come back later, but basically this is saying is that DNA is the genetic material. What does genetic material mean? It means the information to make proteins is how we're going to simplify that down for right now. We'll talk more about how it does it later, but the main purpose is to make proteins, and that's what we mean by genetic material. Now there are some exceptions in viruses, but as you'll see in the viruses unit, they're not considered to be living. And so some viruses use RNA as their genetic material instead. So there are some viruses that use DNA and some use RNA. But all of our technical living things in the 3 domains of life all use DNA. Standard 2 is, well, what is DNA made out of? So our monomer, or our single part, is called a nucleotide. Our nucleotide is a little fancier than some of the other ones we've learned. It has 3 pieces. It has a circle, a pentagon, and a rectangle, and each of these makes up one nucleotide. The sugar, is the pentagon right here, and this is a pentose sugar. Pentose sugar. It could be either ribose in RNA or deoxyribose in DNA. The circle is a phosphate group. The same phosphate groups we found in ATP. The other part of this, the rectangle, is a nitrogenous base. And so we would use those to represent our nucleotides. Standard 3 is all about, well, how do we then link those together to make polymers? Well, we make what's called a backbone. And it's specifically a sugar phosphate backbone. So the main structural component is the sugars and the phosphates. And they run along the sides. So it looks like this. I hate drawing pentagons, but you'll see that it's bonded like this over and over again. So you take those monomers and you make them into polymers. And so we have a sugar phosphate backbone. Let me do one more. Now with this one, you take the monomers and you bond them together the same way you do all monomers to polymers through condensation reaction, and you're forming covalent bonds. And so we have, in this example here that I've drawn, 2 new covalent bonds. So these bonds are nice strong bonds, and when we formed them, we must have gotten water out, right? Would have been a condensation reaction. Oh, and that is actually standard 5, so I'm going to write that down right here. So when we form these polymers, we do a condensation reaction. So this condensation reaction releases water as we link them together to form our polymers. And so specifically this happens in both RNA and DNA. The reason we think of it more in terms of RNA though is because RNA is single-stranded. And so we see when you link them together like this, you're just done. That's it. Alright. Standard 3, backbone, covalently bonded, great. Alright, standard 4 says that the bases are what form the code. So the bases are adenine, thymine, cytosine, and guanine in DNA. And then in RNA, instead of thymine, we have uracil. And what we do with these is we look at the order of them, and that is what tells us the information. So if we abbreviate it, it'd be like A, T, A, T, A, T, 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 A, C, G. And then your body interprets this as a code. So these nitrogenous bases, you need to know the full names, but then we abbreviate them when we write them out. These are what actually form that information that we were talking about in Standard 1. Standard 6 is all about the DNA double helix. And so we mentioned this already, but instead of it just being the single strand like before, now we have those nucleotides, and then we have they're hydrogen bonded to another strand, like so. That's a really bad upside down pentagon, sorry about that. I'm just going to do 2 for this one because these are a lot harder to draw. There we go. Alright, and so we can see here that they're running in opposite directions, right? So this one, you have the phosphate on the top, and on this strand the phosphate's at the bottom, because they're running in opposite directions. We call this anti-parallel. Anti-parallel. So the way they're running is anti-parallel. You do not need to be able to draw the three-dimensional helical shape. You can always draw it flat like this. Now they're linked in the middle by hydrogen bonds. We can tell they're hydrogen bonds because they're dashed or dotted lines instead of the solid lines that we use for covalent bonding. You need to know that adenine bonds to thymine and cytosine bonds to guanine, and vice versa, right? So guanine to cytosine. These are reversible. Now, if you are in SL, that's all you need to know. If you are in HL, you need to know the number of bonds. Okay? So for HL only, you need to know the number of bonds. And the number of bonds are adenine to thymine is 2, and cytosine to guanine is 3. 2 H bonds versus 3 for cytosine and guanine. Alright, standard 7. We talked about a lot of these already, but standard 7 is our DNA versus RNA comparison table. Our DNA versus RNA. So first we have the number of strands. In DNA we have 2, RNA we have 1. The types of nitrogenous bases, so our bases, we have A, T, C, G versus A, U, C, G. And then the type of sugar. And you should be able to sketch out ribose and deoxyribose. So I'm going to do that quickly, and I'm mainly just going to highlight the difference. So we have our carbons, and we number them like a clock. So you start, the O is the 0, and you go clockwise. So the first bend would be 1, the second bend would be 2, and then you go around like that. And so on deoxyribose in DNA, you only have a hydrogen on the second carbon. Whereas in ribose, in RNA, when we do the bend, the second one, you have a hydroxyl or an OH group. So there is an oxygen here. So deoxyribose, no oxygen, deoxy, and then ribose has the oxygen on that second carbon. DNA is the genetic information, and then RNA we'll learn more about later, but there's mRNA, tRNA, siRNA, rRNA. There's over 200 types of RNA. There's one type of DNA, there's over 200 types of RNA, and we'll learn more about those in future units. All right, 8. Oh, complementary base pairing. And so complementary base pairing is, I forgot to tell you earlier, but complementary base pairing is when the bases complement each other and they go together. And we already know what these are. It's A to T and C to G, or A to U in RNA. And so this complementary base pairing, since they always match like this, what that means is you can replicate it and make more of it over and over again because you just pair them up correctly when you're adding new bases. Or you can express it and turn it into RNA. And so replicating would be making more DNA, expressing would be making RNA. And so you can take those complementary letters and know that it matches up. For example, right, if I tell you the sequence is A, T, C, G, you know that the matching one in DNA would be T, T, A, G, C, and so does your cell's machinery. And it can take this apart and make 2 new strands from this over and over and over again. So it allows for it to be replicated over and over and over again, and this is based off of those hydrogen bonds between the bases, right? Hydrogen bonds between bases. For standard 9, is just kind of one of those ones that are like, hey, know this. And it's basically saying that because there are 4 bases and you have so many like base pairs, you have basically an infinite number of combinations that you can make. And so this is one of the useful things, right, is humans have 2 billion roughly base pairs, which means if it's like 4 to the x and x is 2 billion, you can see that that is a lot of different combinations that you can get. And so that means we can have all of our different proteins and all of our different traits encoded in the DNA. This is also why some scientists are looking to use DNA as a storage device for computers in the future. So that way, since it's more choices than binary, right, there's 4 choices, that means there is significantly more possibilities in the number of the amount of information you can store. And DNA can be stored in a cell that's, microscopic. So if we can figure out how to efficiently do it, we could create like unlimited storage capacity. So it's pretty cool. Standard 10 is the last SL standard. It says that it's universal across life. The genetic code is universal, and so are the bases, and so is DNA. Therefore, there must be common ancestry. The logical and non-random explanation for how they all ended up with the same genetic material that's interpreted in the same way is because it was passed down from a common ancestor. So that is the end of SL. Now we have our HL standards. Standard 11 is something that we could see on the previous image. It's the direction of RNA. And so we talked about this briefly already, but basically when you draw your molecule, specifically your sugar, each of the carbons gets a number, right? And so this would be the first carbon, so you start at the 0, you go clockwise around, 1, 2, 3, 4, 5. And the carbons matter, except for carbon number 4, we don't care about that one. Carbon 1, this is where the nitrogenous base attaches. Carbon 2 tells you if it's ribose or deoxyribose, in the other order, ribose, deoxyribose. Carbon 3 is where you make the polymer to monomer linkage. So this is where the new one hooks in through condensation. And then carbon 5 is where the phosphate links in. So carbon 5 is where the phosphate links in. So each of these carbons matters. Now you'll see, right, whenever we draw it, we always draw it typically at the 5' end up, but since they run anti-parallel, they're in opposite directions. And so it matters. We talked about this briefly in class, but this phosphate here is actually not a phosphate when you have free-floating nucleotides. It's actually a triphosphate, and it looks like this. You chop off those phosphates, you get energy out, and that's what allows for condensation reactions to happen. Very much like ATP, the removal of phosphates creates energy, usable energy, that allows it to form this condensation reaction. So that's why, if you're like, well, why does it hook into the bottom like that? That's why, because that's where the high energy phosphate is coming in, and then it can attach in like that. So you always build by adding to the 3' end, so you're building in that direction. Standard 12. 12 is our new word, so we have our purines and our pyrimidines. Our purines are A and G, and these have a different structure. They have a 2-ring structure compared to our pyrimidines, which are C and T, with their 1-ring structure. I got to give a shout out to one of my students. I got to draw our cow to help us remember why they are the way they are. There you go. You got to add the smile. So remember, when we think of A and G, we think of ag or agriculture. That, of course, makes us think of cows. And so our purines make pure milk, ag, and then cows have 2 horns. That's how you can remember purines. Pyrimidines are our cats, right? Cats live in the pyramids of ancient Egypt, and they have their collars, and their collar represents the 1 ring. Okay. And so, yeah, that's how we can remember pyrimidines. This goes like this. A bonds the T, C bonds the G. Because there's 2 rings and 1 ring, 2, 1, 1, 2, they always have the same length. And since they have the same length, that increases the stability of the molecule. So DNA as a double helix is more stable, but the reason it's more stable is because that A, T, and C, G are the same length, which gives it the same 3D structure, and you don't get any warping or bending of the backbone. Right, so this is increasing the backbone stability. 13, nucleosomes. So how can we fit a DNA strand that is 2 meters long into a cell that is only like 50 to 100 micrometers, right? So this is way smaller. This is times 10 to the negative 6 meters, but it has something in it that's 2 meters long. We've got to coil it really, really tightly, and we call that a nucleosome, which is really hard to draw 3-dimensionally. But there's 8 proteins, or an octamer, of 8 histone proteins. Those 8 histone proteins have DNA wrapped around them, and then a linker histone protein. So a total of 9 histone proteins, 8 in the middle, 1 on the outside, and then the squiggles are the DNA. Right? So the DNA is bound to this, and this is for storage, and it regulates expression. Regulates expression, which we said was making RNA. Because if it's all linked up like that, you can't do it. All right. And you should be able, we use the molecular visualization software JMOL to look at it. So you should be able to look up JMOL and see it as well. All right, standard 14, Hershey-Chase experiment. So the Hershey-Chase experiment has nothing to do with chocolate, unfortunately, but it has everything to do with how did we determine that DNA was the genetic material. Right? So we know now it's the genetic material, but when they were investigating genetic material, they needed something with repetition or repeats that had variation. And both DNA and protein fit this criteria. Right? They have repeats and variation. This one has 4 letters, and this one has the 20 amino acids. So they were like, which one is the information? And so what they did was the Hershey-Chase experiment. And this is a good example of something that might end up on like a paper 1. So make sure you know this one. And so the Hershey-Chase experiment, they had radioactive phosphor and sulfur, or radioisotopes of phosphor and sulfur. And they took these, and they looked to see which one they found when they used a bacteriophage. Which one they found when they used a bacteriophage. And so they took the phosphor 32 and the sulfur 35, and they grew bacteriophages in them. So a bacteriophage is a virus of bacteria. A virus of bacteria. And it looks kind of like this. Looks kind of like a spider almost. It has the DNA inside of it, and it has a protein capsule. So it's made out of protein on the outside and DNA on the inside. And so what these do is they come and they find a bacterial cell and they inject the DNA inside of them, like that. And then it uses the cell to reproduce. So what Hershey and Chase did is they did this, and then they swirled it around in a centrifuge tube, and they found that there was lots of extracellular sulfur, but very little low amounts of extracellular phosphorus, because the phosphorus is found in DNA. So phosphorus is found in DNA, whereas sulfur is found in protein. And so you can see by looking at this picture, the DNA is in the cell, so it's not extracellular, but the protein capsule, once it's been used, it just kind of floats there in the extracellular matrix. So they're like, oh, look at all this. So they're like, well, if this is what went inside, it must be the genetic material, because it went inside the cell to make more copies. So at the time, they already knew how viruses reproduce. They knew they reproduce using other cells. And so they're like, oh, okay, this is what did it. And so using radioisotopes, didn't write that down, radioisotopes, they were able to figure it out. So remember, radioisotopes have different number of neutrons than your normal ones. All right, our last thing we need to know is another thing that you could show up on paper 1 or paper 2, and that's Chargaff's data. Chargaff was a scientist that was like studying DNA structure, early DNA structure. And originally they're like, oh look, we have all of these adenines, cytosines, and guanines. We think they look like this kind of like weird thing. And so it kind of was like a square where you had like a base off of each corner. And they're like, look, this is what it looks like. There's 4 of them. They must repeat like this over and over again. So they thought there's like, it would look like something like this, right? Oops. You'd have a whole bunch of these. And you can see that if this was the case, and you were to sample, and you were to find the percentage of adenine, thymine, cytosine, and guanine, it should be 25, 25, 25, 25, because there's 1 of each on every single nucleotide. This is the tetranucleotide hypothesis. Well, Chargaff went and collected some data, and he did not get that. He got like 29, 29, and then, oh, I shouldn't have just made these numbers up. 58, 16, 16, there we go. And so you've got, okay, that's not the same as 25 and 25. And he did it for a bunch of different organisms, and he kept getting that it was never exactly 25, 25, 25, 25, because it wasn't. Because this is not what it looks like, right? We know what it looks like now. And so you can have varying amounts of A, T, C, and G, depending on what your sequence is. So if you have more AATs in something like this, then you'll see that the ratio is higher for A and T than it is for C and G. Now, what do we do with this information? Well, the other thing he found is that the amounts of pyrimidines was equal to the amounts of purines. So remember, we've got our cat and our ag, so these 2 are equal. Well, then he continued to investigate, and he found that not only are these 2 equal, typically T and A are equal, and C and G are equal. So these 2 are equal because they're bonding to each other, and then if you think about it, right, if you've got C here and G here, and then T here and A here, those, and you add them together, they must also be equal by math. It has a property name, but I can't remember what it is. And so because of this, this is what he found. Oh, the pyrimidines are equal to purines, which it should have just been all 4 were equal, but that was not true. So he was like, wait, hold up, this doesn't match. This is another example of something that could show up on paper 1. All right, so he found this. Purines and pyrimidines are equal, and then T is equal to A, C is equal to G. They have ratios that are close to 1 when you compare them. And that is the A12 standards.